I can see Joe, and there's Roy. So we have a double act for our last talk of today. I think Joanne's going to be driving the presentation, but we start with Roy Biggs. So Roy is representing the Poultry Industry Association, and Joanne is another of my colleagues from ESR. And they're going to talk to us about on-farm and in-processing studies on Campylobacter prevalence. So Roy is a food technologist with experience in senior management in the food industry. For the last 24 years, he's been in technical management roles in the poultry sector, followed by consultancy in New Zealand and internationally. I have to tell you this, Roy is the only New Zealander to be presented with the International Leadership Award from the International Association for Food Protection, which is the largest group of food safety professionals in the world. So we're very glad to have Roy with us today. And Jo Kingsbury, Dr. Jo Kingsbury, is a food microbiology senior scientist, and she's in Rob Lake's Risk Assessment and Social Systems Group at ESR in Christchurch. And her work focuses on detecting, describing, and understanding the risk of disease-causing microbes, such as Campylobacter and Salmonella, in food and food production environments. She's recently co-authored reports addressing the potential for transmission of SARS-CoV-2 via food or contaminated packaging for the New Zealand Food Safety Science and Research Centre. And I believe, Joe, that you're right in the process of updating a more recent publication addressing Delta for that COVID work. So welcome both to Roy and Joe. I think Roy will start us off.
Firstly, thank you for inviting us to present these two projects. We certainly enjoyed working on them. Hopefully you'll enjoy hearing about them. So PIANZ, the Poultry Industry Association, is a representative body for the industry, and the veterinary technical group within PIANZ, supported by the CEOs of the major companies, which is the PIANZ board, have agreed to cooperate in terms of the control of zoonoses. So the instigation of these two projects are the result of that cooperation. Next slide, please, Joe. Sorry, I was trying to move it then. Next slide, please. Oh, thank you. Oh, got the notes on there as well. Yeah, it's okay. It's okay. There's some notes attached to the slide. So in the early 2000s.
Oh, sorry, Roy. Are you seeing the proper slides or are you seeing the notes?
No, it's all right. I got the notes on another screen in front of me, so I'm fine. I can see the presentation. So in the early 2000s, New Zealand had one of the highest rates of Campy in the world, reaching a peak of 380 per 100,000 people in 2006. Following the identification of poultry as the most important contributor, and interventions by the poultry industry, mostly at the processing level, resulted quite rapidly in a 50% drop in cases, as you can see from the nice little red lines on the slide there. So next slide, please, Joe. So since that time, the poultry industry have been working together with MPI sorry, to reduce Campylobacter numbers. There's been a more gradual decline in incidence in recent years, despite an increase in poultry consumption per capita. So you can see the decline in terms of the line below the MPI target there. We've done pretty well. Chicken consumption's been going up in the next graph. And then the blue line on the graph on the right is the mean log count. The limited detection of this method used then is 2.3, so you can see we're below the limited detection. And the red line is the prevalence of positive samples. So you can see there's been significant improvements. We've got a lot of slides to go through, so I'll talk fast. Next slide, please. This project, the first project, involved a longitudinal survey following one broiler flock from its parent breeder flock through to processing, looking at the role of reservoirs and vectors for Campylobacter colonization of the flock. It takes a lot of people to deliver a trial of this size, and you can see some of them, at least listed on there. The picture at the top of there you all recognize, I'm sure, Mount Taranaki with the farm we use, Campy farm, 9 sheds of 40,000 birds each below it. Next slide, please. The majority of Campylobacter controls which we've implemented have been in processing, because that was the quickest way to do it when we got the ultimatum from MPI in 2006 that we needed to do something. We would prefer to reduce broiler colonization on farm if we can find a way of doing it. We've done that previously with salmonella and it worked very well. But with previous experience from work done with Listeria, the industry recognized the potential of whole genome sequencing for linkages between Campylobacter in poultry with the contamination sources and vectors. I'm going to hand over to Joe to do the real science.
Thanks, Roy. Sorry, my slides are not progressing properly. Okay. And thanks also to Libby for the introduction. So the broiler farm longitudinal survey followed a single broiler flock from the breeder flock through hatching, through the whole life of the flock till the birds went off for processing. And we took multiple samples of multiple different sample types along the way, including potential vectors for a Campylobacter ingress into the sheds, for example, insects, rodents, the workers on the farm, and the catchers and crew that came in to catch the birds to send them off for processing. We also took samples of potential Campylobacter reservoirs on and off the farm, for example, the breeder flock, the flock that was in the shed before our study flock, and a control flock that was raised at the same time as our study flock. And we looked for evidence of flock colonization along the way, for example, cloacal swabs of live birds and cecal contents and rinsates of dead birds or birds at processing. All of these samples were sent to Marie Callender's lab at Tegel, New Plymouth to test for Campylobacter. The first step of this involved enrichment broths. Any samples that tested positive for Campylobacter, Marie sent to 2 isolates to Anne Midwinter, Patrick Biggs and Nigel French at Nassau University to test by whole genome sequencing. And she also sent aliquots of the enrichment broths to Anne and Patrick to test by a novel meta-barcoding method that Patrick developed as part of this study. And then the overall goal is to link Campylobacter from reservoirs and vectors to what's colonizing the poultry. This is a sampling timeline for the study. We have the sample categories along the side, timeline along the bottom, and where you see a red box, that means at least some of the samples tested positive for Campylobacter. Green means no samples tested positive. So some of the key findings were the breeder flock tested positive for only Campylobacter larvae. So we didn't find this anywhere else in the survey. This is at the time of the laying of the eggs for the study flock. The flock that was in the shed before our study flock tested positive for Campylobacter just before depopulation, and the shed environment was positive as well. But after depopulation, the shed was cleaned out and disinfected, and we didn't detect any Campylobacter then. The study flock was placed as day-old chicks into the shed, and then every 5 days we tested for evidence of flock colonization and also samples from the sheds inside and outside the sheds. We found no evidence of flock colonization until after the first cut, which is when the first lot of birds get sent off for processing. At this time, about a half of the catcher samples tested positive for Campylobacter, but we didn't see any Campylobacter in the ceca of the birds that went off for processing. 1 out of 15 rinsates tested positive, but this could have also happened by cross-contamination. But after this time, we see colonization of the flock and more positives in the shed environment. So in total, we tested 738 samples and 30% were positive for Campylobacter. The species we isolated the most was Campylobacter jejuni, and this was the only species really that we isolated from our study flock. So we only did whole genome sequencing on C. jejuni, 199 isolates. This is a minimum spanning tree representation of the whole genome sequencing data that was produced by Nigel French. So the objective is to link what we're seeing in colonizing the birds with potential contamination sources. If you're not familiar with this type of diagram, every circle represents one or more isolates. The bigger the circle is, the more isolates are indistinguishable by the typing method. The branch lengths represent the number of alleles different between the isolates. And the different colors represent the different sequence types. So you can see there are 7 sequence types among the 199 isolates. The most prevalent was 6964, about 2/3 of them were 6964. And this is the most commonly isolated sequence type from poultry in New Zealand in recent years. And you might have heard Nigel talking about this as a fluoroquinolone and tetracycline resistant sequence type. So about 2/3 of the isolates from the study flock had this sequence type, and many of them were quite highly clonal. And interestingly, we find some of these were indistinguishable from isolates from the previous flock that was in the shed, as well as the control flock raised at the same time. So this suggests a potential on-farm reservoir of this sequence type. But we also saw indistinguishable isolates from the catcher, crew and equipment, so that might be another contamination source. And similarly, about 1/3 of the samples from the birds were ST50, and some of these were indistinguishable from catcher equipment as well. One of the sequence types, clades of ST40, was only isolated from wild animals outside the shed, so we don't think they were potential contamination sources. And one of the clades, each of ST45 and ST50, were only isolated from catchers and equipment. Another key element of the study was the development of a Campylobacter-specific meta-barcoding approach by Patrick Biggs at Massey with help from Anne. And what this would look like is you take your environmental sample. In our case, it was an enrichment broth of the environmental sample. You extract the DNA, and then you amplify up a specific genomic region that is present, and it's specific to Campylobacter, but there is a lot of sequence heterogeneity across that sequence that ideally would correlate with what sequence type it is. And so part of the study for Patrick was deciding which genomic region to target. You then perform next generation sequencing on the amplified region, compare the reads against a database to then assess the Campylobacter allele types and proportions from each of the different sample types to make linkages between what's present in the flock and potential contamination sources. So in this made up example, you see that chickens have the blue sequence type, and this matches what we see in flies, but again, this is just a made up example. But what Patrick found is that the meta-barcoding method really proved to be a complementary approach to the whole genome sequencing, but it also uncovered quite a few new and interesting insights. Using the method, he was able to detect most Campylobacter sequence types that he did by conventional isolation, but also some sequence types that were never isolated. So we only sequenced 2 isolates per positive sample, and the meta-barcoding method has the potential to detect multiple sequence types. He could detect Campylobacter from samples at earlier time points than he could by isolation. For example, he detected 6964 from the shed samples following the clean out of the previous flock. So this is supportive of our on-farm reservoir contaminating our broiler flock. But the method, the meta-barcoding, doesn't differentiate between live and dead cells. So it is still possible that these Campylobacter were not live. He could also detect 6964 from the live chicken samples when the chickens were only 10 days old. So this is a lot earlier than what we detected by isolation, which wasn't until the birds were 35 days old. So this again supports the on-farm hypothesis for contamination. This is obviously before the catchers were in the shed. And he, another interesting finding was that he could detect 6964 from the breeder flock as well. And this was at the time that the eggs were laid for the study flock. So this is, we only isolated Campylobacter lowri from the breeder flock. So this was a really interesting finding. And he also identified a wide range of sequence types from capture samples, which is consistent with the whole genome sequencing results. So to conclude, we've identified several potential contamination sources for the broiler flock in this study, but in a literature review that Nikki King did as part of this study, she determined that reservoirs and vectors for Campylobacter contamination of flocks do vary to some degree by farm location, seasonality, and housing system. So that makes it really difficult to control the Campylobacter in the broiler flocks. But we hope that some of the findings that we get from this study should help the poultry industry to develop some on-farm risk management practices. One example might be a focus on shed cleanup, but what the study also does is raise some other interesting areas for future research. For example, it would be interesting to do the same sort of study in a different housing system, for example, free range. But I'm going to move on to the next project that was commissioned by PIANZ. So we are now moving from the farm to the processing. This was commissioned in 2020, and the majority of the work was carried out this year. Some of it's still ongoing. The project was also co-funded by MPI. The study team are involved, and I'd also really like to acknowledge all the other people listed that also played really important roles in the study. So the survey design involved sampling different steps along the primary and secondary processing at processing plants of the three biggest producers of broiler poultry in New Zealand, anonymised plant A, D, and F. And we sampled carcasses from 8 different primary processing steps and from 7 different types of products at secondary processing. We took 5 samples of each type at each time for a total of 75 samples per sampling event, and we sampled each plant 6 times. So it was a total of 1,350 samples. The sample types were rinsate. So we or Roy or in the samplers put the carcasses and the portions into bags with buffer in it, rinsed this around for 2 minutes, and then sent that rinsate off to Marie Callender's lab at Tegel New Plymouth Lab to enumerate for Campylobacter. And Marie also sent isolates from the samples to Anne and Patrick at Massey University for whole genome sequencing and analysis. And the aim is to establish the numbers of Campylobacter at each step and determine the impacts of the processing steps on the numbers and types of Campylobacter present. So this is some of the data from the primary processing sampling. We're breaking the data down by the different 3 plants here. The Campylobacter numbers in log are on the y-axis, and the different areas for sampling are along the bottom. And this is the order of the processing line. Some of these processing steps have the potential to increase the Campylobacter contamination, for example, if there's fecal spillage from the process, whereas some of them have antimicrobial steps involved. And the most important antimicrobial processes are the spin chill, which involves a chlorine wash, and the acidified sodium chloride step, which is the end of the primary processing. So we have approximately 7 log, this is a violin plot if you're not familiar with these. So the line in the middle is the median observation. The width of the violin corresponds to how many samples had that observation. So we go, when the chickens come into processing, there is a median of 7 log CFU colony forming units on the carcasses. But by the end of primary processing, we see a 6 log reduction in numbers. And in fact, 76% of the samples at the end of primary processing had no detection of Campylobacter, so less than 20 colony forming units per carcass. And we're also exploring plant-to-plant differences at each step. So, for example, you see a greater reduction between the pre-scold and post-scold steps at plant D than what we see for plant A and plant F. And so we are considering those results against the different types of processes that the different plants have. And Beverly Horn and Bridget Armstrong are currently using this data to create a quantitative risk model so that you can predict the numbers of Campylobacter on product by making different changes to different processing steps. A similar study had also been conducted in plant D back in 2013, but since then there's been a lot of improvements made to within the plant. And so we compared our data against the 2013 data. You can see that at the beginning of processing, there are similar numbers on the carcasses, but at the end of primary processing, there is significant differences between the Campylobacter numbers. There is a 4-log reduction. There was a 4-log reduction in numbers in 2013, 6-log reduction now in 2021. So that really indicates that the changes to the processing is making an improvement to detection of Campylobacter on the carcasses. And Roy has mentioned before that this isn't just specific to plant D, this is also going on in other plants as well. Now we're looking at numbers on secondary processing. So again, broken down by the 3 different plants, and on the x-axis broken down by the different portion types or sample types at secondary processing. So one of the first key findings that we see is that the numbers on whole birds in plant F were a lot higher than what we see in plant D and plant A. And they're also higher than what we saw from plant F at the end of primary processing. And we think that this is related to the fact that plant F has separated primary and secondary processing plants, whereas in plant A and D, the birds are going directly from primary to secondary. At plant F, they are travelling and it's taking several hours to go from primary to secondary. So it could be that the birds are bumping around in the bin, and maybe some Campylobacter coming out of the feather follicles that are being missed in some of the other in plant D and A. A second key finding is that the drums have much lower Campylobacter numbers than any of the other portion types. And one possible explanation for this is that the birds at primary processing are hanging by their legs. And so any contamination is flowing down from south of the legs. So they're not, there's no cross-contamination of the drums there. And another interesting finding is that the Campylobacter has been reported to be present on the skin, but not on the meat. It's present within the feather follicles particularly. And so we might expect that when we remove the skin, we reduce the load of Campylobacter. But this isn't consistently what we see. For example, in plant A, when we're removing the skin, we're actually seeing increasing loads of Campylobacter. So this might be consistent with some cross-contamination going with the deskinning process. So now I'm going to move back to you, Roy, for industry perspectives.
Thank you very much, Jo. I understand that now, it's good. The new methods introduced in these trials have been the key to the industry getting the outcomes that we wanted. So we now have a much greater understanding of what's going on farm and how the sheds originally get infected. And the meta-barcoding was a real eye-opener for that. The processing plant longitudinal data has already provided data that we're using in other plants that have Campylobacter issues. It gives us a benchmark, a really valuable benchmark. And the insights on MLST types, which we haven't shown you today, there wasn't time, of Campy in specific factories was somewhat unexpected, but useful. So we've also developed new ways of sampling and testing. We had to get the results. So massage sampling using a centrifuge method to get the limit of detection on carcasses down from 200 to 20. So we've done a lot of learning along the way, and we've certainly learned a lot from our scientific colleagues. Next slide, and back to you, Jo. Or do you want me to comment?
That's it.
Yeah. And really thank you to, all the people involved. There's a massive number of people involved in these two slides, both from the sampling, the laboratories, ESR, Massey, and the FSSRC. Brilliant. Thank you all very much. I think we've got some really useful data for the industry to use.
Yeah. Thank you, Roy, and thank you, Joe. That was a really interesting presentation. I've seen it before, but I learn more every time I see it. So thank you. And I'd also like to comment on the incredible collaboration across industry association and researchers, and great to see a co-creation of a presentation from the two of you. So thank you. Clearly, the outcomes of the study couldn't have happened without all of those participants, including those collecting the samples at the poultry site. So thank you, everybody. Now, I do have one question. Well done, Peter. He scored a hat trick. Peter Cressy, seems like all in, sorry, I'll start again. Seems like all in, all out, depopulation of sheds may have benefits. Is this practical from a supply perspective?
I did work this out, Peter, as one of the many outcomes of this trial. Unfortunately, we would have to have a lot more sheds, somewhere around $160 million worth. So as soon as I said that to the industry, they seemed to lose all interest in that concept.
Great. Anybody else got any questions? I have one for you, Roy. We talk about, with COVID-19, we've all learned the language of diseases, and we talk about elimination. Is there a goal to try and eliminate Campylobacter, or is that the holy grail?
That would be the holy grail, but if I give salmonella as an example, we went down from 17% prevalence to the last 5 years, it's been less than 0.2% prevalence. So you can get close. Now we're nowhere near that level on Campylobacter, but we do think that with the outcome of this trial and other information we continue to get, that we can get a little bit better.
Good. That sounds good. And it looks like industry and the workers and the researchers are all there wanting to support that holy grail. Okay, well, I've got two more questions. Sorry, got to keep an eye on this. So I have an anonymous attendee who asks, is MPI lowering the prevalence limits again on Campy?
Is that an anonymous attendee from MPI, do you think? But they were lowered in July this year, both prevalence limits and the number of birds you have at the enumeration limit. So, and that was significantly lowered from prevalence limits was 30%. That's gone down to about 22%. The number of samples above log 3.78, the limit used to be 6, it's now 2. So hopefully, well, I know they will lower them again, but hopefully we get a little bit of breathing space to learn out how to do it.
Yeah. Okay, thank you, Roy. And we have a question here from Craig Shadbolt, who's rapidly chasing Peter Cressy for questions. Is there likely to be a best bang for buck intervention at farm level, or does this vary by farm operation, housing system, etc.?
We believe there's quite a lot of variance, and we know there's a significant difference between the barn raised method of production and the free range. And the targets we have for barn range first cut seeker, so when they come to the processing plant, they seek out and look at them, is 35%, which most plants are meeting. The free range target is 70%, which most plants are not meeting. Free range really does make a big difference.
Okay, thank you. So those are all the questions. So I think we were scheduled to finish at 3 o'clock. So I just want to, before I say thank you to people, I'd like to make an announcement on behalf of the Food Safety Science and Research Centre. So on the 20th of October, so just under a month away, at 10 o'clock, 20th of October, 10 o'clock, distinguished Professor Nigel French will be giving a webinar for 1 hour on emerging infectious diseases. So put that date in your diaries. I don't have the exact time. Oh yes, I do. 10 o'clock, 20th of October. I think given these times of pandemic, it's a very relevant subject for us all to participate in. So I'll finish with another whakatoki to end our korero, but I'd like to thank all of the presenters today who did a fabulous job and wrestled the technology and joined us. Over 130 people at the beginning of the session. So thank you to all the participants, all the listeners, and thank you for your questions. And I'll just end with this whakatoki. Ki te kotahi te ka kaho, ka whāti, ki te ka puie e kore e whāti. And that means is if there's only one toi stem, it will break. But if they are together in a bundle, they will never break. We're stronger together in everything we do. So I think that's quite clear from the presentations we've seen today. So go well, everybody. Kia kaha.